Cells form the basic units of life. We know this. And by now, we even know how the discovery of cells, as seen in the tree cork, was a breakthrough in biological studies. Now it's time to have a look at how the cells actually look under a microscope. For this, we select onion peel as our specimen. But before we begin with the procedure, let's first collect the necessary requirements. We will need a microscope, a slide, a cover slip, iodine solution, water, and forceps. Anything else we're left with? Well, the most important of all is the specimen, that is an onion peel. Now that we're all set with the equipments needed, let's begin with the amazing experiment of observing the onion cells under the microscope. Firstly, we take the onion peel and with the help of the forceps, pull away the thin lining from the inner surface. Before placing this on the slide, we first add a drop of water on, then place the onion peel on it. We are not supposed to touch the peel and even the slide with our fingers. Next, we add 2 drops of dilute iodine solution. Hold on, do you know why is iodine solution added to this specimen? We do this so that we can clearly see all the parts of the cell. Else it's difficult to study them as they are transparent. It would even be difficult to differentiate between the various parts. Now we place a cover slip in this manner and make sure that no air bubbles are formed on the slide. With this, our slide is ready. This entire process of setting up the microscope along with the slide is termed mounting. It's also important to see that there is enough light falling on the mirror of the microscope. Shall we observe it now? Here we go. Wow, doesn't it look amazing? Now before studying these, tell me one thing. Do plants and animal cells look the same? Do they have the same organelles? Or are the two types different in some ways? Well, plant and animal cells are slightly different. When cells from a human cheek are scraped with a sterile cotton bud and mounted, similar but not exactly same cells are seen under the microscope. Roughly, we have these thick, distinct borders that differentiate each cell from the other one. Then we have these thin, tiny dark spots scattered all over. Also, we can spot this prominent circular structure here, which is very darkly stained. Any ideas what these things are? Well, these are the cell constituents. Let's get introduced to each in the next video. This is where we left off in the previous video. We mounted plant cells and animal cells on slides to observe them under the microscope. Let's study them in general in this video. For our convenience, let's select just one. Say the onion peel cells to understand the structure and the contents of the cell. Roughly, we've seen these thick, distinct borders that differentiate each cell from the other one. Then we have these tiny dark spots scattered all over. Also, we can spot this prominent circular structure here, which is very darkly stained. Any idea what these things are? Well, these are the cell constituents. Let's get introduced to each. This outer margin seen here is called the cell wall. If we zoom in a bit, we notice that there is another layer in the inner side of this cell wall. This is called the cell membrane. Yes, the outer wall is the cell wall and the inner layer is called the cell membrane. The cell membrane is also referred to as the plasma membrane. It is porous in nature. So what does porous mean? It simply means that any material can move in and out of the plasma membrane. But isn't that dangerous? Of course it is. But the simple solution lies in the nature of the plasma membrane. That is because it is selectively permeable. So what does selectively permeable mean here? Just like a guard standing at the office door who allows only members with the identity cards to enter and exit, the plasma membrane also allows only specific materials to move across. We will look at this in details in our upcoming videos. This was just to understand how the plasma membrane is selectively permeable. Now let's move ahead to know about the cell wall. Do you think all cells have cell walls? Well, the answer is a no. That's because animal cells do not have cell walls. An animal cell only has the plasma membrane and not the cell wall. The cell walls act as protectors for plant cells. It's composed of a non-living rigid structure that is cellulose. It not only acts as a protector, but also gives a proper shape to the plant cells. It also helps in cell-to-cell interactions and provides a barrier to undesirable macromolecules. It protects, it gives shape to the cells, and it helps in cell-to-cell interactions. Clearly, the cell wall is a very important part of the plant cell structure. Now let's get into this lighter region here. This lighter region is the cytoplasm. It's a jelly-like substance filling up the cell space. And these tiny parts in the cytoplasm are the organelles. And what exactly are they? Just like the human body has different organs, the cell has various organelles. And just like the organs, each organelle performs a different function. Let's study each organelle and its functions in our upcoming videos. If we are given this image and asked what exactly this is, I'm sure your answer will be a cell. Now to study this, we need to study every component present within. And not only within, we even need to study the covering, that is, this part in detail. Now can you tell me what exactly is this named as? That's right, it's called the cell membrane or the plasma membrane. In our previous video, we have learned that the cell membrane is selectively permeable in nature. That is, it only allows specific materials to enter and leave the cell. Any guesses what is the plasma membrane made up of? The plasma membrane is made up of lipids and proteins and also has some carbohydrate chains. There are two things that freely move in and out of the cell. Those include gases and water. Do you think these molecules need energy for traveling across the membrane? Let's take a simple example to understand what exactly happens in this case. We know what happens when a drop of ink is added to a glass of water. The single dark-colored ink drop spreads when added to water and the intense blue color fades slightly. If we see at the molecular level, then the ink molecules, which are compactly packed in the single drop, get a chance of dispersing in a larger area when added to the water. This is nothing but the process of diffusion. And we know that this dispersal process does not require energy. The molecules just try to move freely in the space available, and as a result, diffusion occurs. The same story is seen when gas or water molecules travel across the membrane. Take carbon dioxide for example. When cells undergo cellular respiration, a lot of carbon dioxide accumulates in the cell. On the other hand, there is very little carbon dioxide present outside the cells. Hence, the carbon dioxide molecules move outside the cells. Now, there is very little oxygen left after respiration in the cells. So the oxygen from outside the cells moves in through the plasma membrane to make up for the lost oxygen. That means carbon dioxide moves out and oxygen moves in. Both the gases move from their area of higher concentration to an area of lower concentration. And this diffusion process does not require energy. Diffusion is one of the processes by which transportation of several substances is carried out across the cell membrane. But it's not the only one process on which the cell relies. There are a few more methods that the cell adopts for transportation of substances. Let's have a look at these processes in the next video. Here's a cell on your screen. We have already seen that the various organelles inside it are compactly packed inside this delicate membrane called the cell membrane or the plasma membrane. We've even seen that the cell membrane makes use of diffusion process to transport gas and water molecules in and out of the cell. However, diffusion is not the only process employed. There are a few more on the list. Let's have a look at these methods in this video. Tell me one thing, how do we transport water from a lower height to a higher height? We need a pump. Because this does not happen on its own. A force or some energy is required to pull the water upwards. Similarly, in a cell, some molecules have to be carried inside or moved outside the cell. This requires energy to be spent. The energy used is in the form of ATP molecules. This is called as active transport. That is, active transport requires expenditure of energy. The transportation is carried out by protein molecules present on the cell membrane. On the other hand, diffusion does not require energy. Hence, it's a type of passive transport. So, movement of molecules without spending energy, as in case of diffusion, is passive transport. Similarly, movement of molecules across the membrane with the expense of energy is active transport. Now let's get to know one more interesting concept named osmosis. Have we ever come across this term osmosis? Well, quite often. But what exactly do we mean by this? Osmosis is defined as the movement of water from higher water concentration to lower water concentration through a semi-permeable membrane. So let's say we have this container which is partitioned by this semi-permeable membrane. Now we add sugar solutions at both these sides. However, the concentration of each is different. Here we add concentrated sugar solution, while here dilute sugar solution. That means the concentration of water in this solution is obviously more compared to this. So after some time, we see water molecules moving from this side of the partition to this side. And how long will this movement take place? It will occur till the concentration of water is balanced, that is, equalized on both the sides. This is nothing but osmosis. Let's learn some more interesting concepts about osmosis and cell transport in the next video.
Have you ever tried comparing your pictures taken at different ages? You would observe that you have grown up when you compare the different pictures. Have you ever wondered how we grow? We all start life with a single cell. Let us find out how our body grows, develops, repairs itself, and becomes an adult with trillions of cells. Cells increase in number by a process called cell division. Every day, our bodies produce millions of skin cells to replace those lost in normal activity. New cells cover any wound in our body. There are two types of cells in living beings. The cells which help in growth are the vegetative cells. Recall how pollen and ovules fuse with each other in a process called fertilization. They are included in the second type of cells, the reproductive cells. In human beings, sperms and ova are the reproductive cells. When a cell is ready to undergo division, it has to go through a preparatory stage. This preparatory stage is called interphase. There are three stages in interphase: G1 or gap 1, S or synthesis, and G2 or gap 2. G1 is the period in where all the organelles and cytoplasmic components are replicated. During the stage S, the chromosomes including the DNA begin to replicate. This results in two identical copies of chromosomes called sister chromatids. The two sister chromatids are attached to each other at a point called the centromere. During G2, organelles and other enzymes required for cell division are formed. The cells spend a great deal of time during interphase and very short period of time in actual division. There are two types of cell division, mitosis and meiosis. Generally, mitotic cell division takes place in vegetative cells and meiotic cell division takes place in reproductive cells. Mitosis consists of four phases: prophase, metaphase, anaphase and telophase. In a microscopic view, we can observe the nucleolus, chromatin network and centrioles in a cell. During prophase stage, the chromatin network changes to chromosomes. Nucleolus and nuclear membrane disappear. Chromosomes become visible, appearing as two sister chromatids held together at the centromere. Cytoskeleton, disembals and centrioles change to star-shaped structure called asters. Spindle fibers begin to form. Towards the end of prophase, the aster move towards the opposite pole of the cell. During metaphase, chromosomes are attached by spindle fibers in their centromere. Chromosomes are arranged at the center of the cell, which is called metaphase plate. Anaphase begins with the degradation of proteins that hold the sister chromatids together. Thus, chromosomes are divided and daughter chromosomes are formed. Daughter chromosomes are pulled to the opposite poles of the cell. This results in equal chromosomal numbers in both mother and daughter cells. Therefore, mitosis can be called equational cell division. At telophase, a furrow forms in the center of the cell. Nuclear membrane reappears around each set of chromosomes. Chromosomes cluster at the opposite poles. Nucleolus reappears and spindle apparatus dissembles. Thus, two daughter nuclei are formed. Finally, cytoplasm divides into two, resulting into two daughter cells. This process is called cytokinesis. Since plants' cell walls cannot be constricted by actin fiber, vesicles form an expanding membrane partition called the cell plate. Similar to animal cells, finally divides into two daughter cells. Do you know why offsprings are not identical to their parents? Let us find out the answer to this question with the process of meiosis that takes place in reproductive cells. Meiosis is the process by which haploid cells are produced from a diploid cell. Here, the chromosomes must be correctly sorted and distributed in a manner to create genetically unique cells with half the number of chromosomes as the original cells. For reduction in the number of chromosomes in the new haploid daughter cells, two rounds of divisions are necessary, meiosis I and meiosis II. As in mitosis, meiosis begins after a cell has successfully completed the G1, S and G2 stages of interphase. During S phase of the interphase, the DNA is replicated, producing two copies of each chromosome called sister chromatids. Meiosis I has four phases, namely prophase I, metaphase I, anaphase I and telophase I. During prophase I, centrioles change to star-shaped structure called asters and spindle fibers are formed. Sister chromatids remain attached at the centromere. Up to this point, the cell looks very similar to mitosis. But two events occur in meiosis that we do not see in mitosis. These two events lead to genetic diversity. During prophase I, a homologous pair of sister chromatids, that is paternal and maternal chromatids, pair with each other, forming a tetrad. Once this structure is formed, the second event called crossing over takes place. During crossing over, a physical exchange between chromosome segments of non-sister chromatids occur. This increases genetic diversity. Prophase 1 concludes when the duplicated centriole pairs move to opposite poles of the cell. As they move, the centrioles extend the spindle fibers, forming the meiotic spindle. In metaphase 1, spindle fibers are completely formed and the sister chromatids are attached to the spindle fibers. Then the bivalents randomly align along the metaphase plate. Due to an independent assortment, this alignment is random and adds to genetic diversity. In anaphase 1, the homologous chromosomes are pulled to opposite poles of the cell. In between the two sets, interzonal fibers are formed. As the centromeres do not divide, the daughter cells will have only half the number of chromosomes. Hence, meiosis 1 is called reductional cell division. Meiosis 1 ends with telophase when the nucleolus and nuclear membrane reappear. Chromosomes again change chromatin network. Two daughter nuclei are formed. With the first nuclear division, the cytoplasm may or may not divide. Telophase 1 is succeeded by meiosis 2, which begins without further replication of DNA in the chromosomes. Meiosis 2 is further divided into prophase 2, metaphase 2, anaphase 2, and telophase 2. During prophase 1, centrioles again change to aster and spindle fibers are formed. Nucleolus and nuclear membrane disappear. Chromatin networks change to chromatids. In prophase 2, asters move to opposite poles of the cell. Sister chromatids are attached to the spindle fibers in the centromere. The spindle fibers align the sister chromatids along the metaphase plate. During anaphase 2, centromere of the chromosome divides. Sister chromatids are separated and pulled towards opposite sides. The entire process ends with telophase 2, as chromosomes again change to chromatin network. Nucleolus and nuclear membrane reappear. Again, furrows separate the two daughter cells to 4 haploid daughter cells. The haploid daughter cells will specialize into gametes, either sperm or egg. These fuse in fertilization to form a zygote, which will grow into a child. The child receives half of its chromosome from its mother and half from its father. As man and woman produce millions of gametes and selection of gametes in fertilization is random, this contributes to genetic diversity. Hence, a child is not identical to either parent. Now, can you state some of the differences between mitosis and meiosis? Mitosis usually occurs in vegetative cells and meiosis usually occurs in reproductive cells. In mitosis, there is only one round of division, but 2 rounds of divisions are necessary for meiosis. Mitosis results in 2 diploid daughter cells, but meiosis results in 4 haploid daughter cells. Unlike mitosis, 2 homologous pairs of chromosomes pair with each other in meiosis. There is no physical exchange of any segment of chromosomes in mitosis, but in meiosis, physical exchange between chromosome segments of non-sister chromatids occur.